Here we go again. This is it's the third episode. I was like, what is it again? No, it's the third episode, and as always, my name is Martin, and with me again we have Emmanuel, my dear little buddy. And say hi, Emmanuel.
Hi, I'm back again.
Great. So, well, what do we have to talk about today, Emmanuel?
We have giant pouched rats and e-noses. I should maybe go be more specific.
Yeah, please. I'm quite
talking
confused there.
about how to detect diseases by smell. So there's a project called Apopo that is detecting tuberculosis with the help of giant pouched rats and also landmines. And now scientists want to recreate this by electronic noses.
Okay, I am intrigued. But okay, well, hold on. You said that they were trying to smell diseases. How can you smell diseases?
It's quite unclear. The scientist doesn't know really how the rats detect it, but tuberculosis is a disease that spreads via the air. So they think that some molecules, either from the bacteria or some molecules that the bacteria releases, the rats can smell. So you use the sputum, the mucus that you cough up, and the rats smell that and can detect if there's tuberculosis in it.
Jeez, okay. Do you even know how they found this out, that these rats could do this?
I think it's widely known that rats has a great taste or great smell.
Smell
Receptors. Or,
yeah, smell.
Yeah. And the founder of Apopo liked to play with these pouch rats. And I think he just created a bond and was curious. I'm not that well read about it, but the founder really liked rats. And in poorer countries, I can maybe just give a short background. In poorer countries, how you detect tuberculosis is with a 130-year-old method, where you use the sputum and look at it under the microscope and try to see bacteria.
Mm-hmm.
But it's quite hard because it's quite easy to miss the bacteria. And also it really depends on the personnel there. And so it's between 20% and 60% hit rate on detecting tuberculosis. But in richer countries, they use different methods. So they use microscope, they grow the bacteria, and also use PCR DNA detection to find it. But in poorer countries, for example, where Apopo is located, they can't use all these methods. So instead, you can use the rats. It's much more cost-effective and it's much more quicker. The rats can detect up to 100 samples in 20 minutes.
Okay.
And they have saved so far 12,000 people who got a negative test result in the microscope.
All right. Oh, well, you mentioned before that though the local population didn't trust is not the word, but they thought that these rats were the rats gave diseases to the local.
The government is quite wary of using rats in this kind of detection because it's a negative connotation on rats, and also it's not a lot of research on rats. So it's much better to use microscope, but also maybe use e-noses, electric noses in the future.
E-noses. Yeah. So what the e-noses are supposed to do is they replicate the smelling
Receptors in the rats' noses.
yeah, exactly. So it's they're trying to copy the rat's own smelling sense.
Exactly. So there's, for example, one where you just blow out on a small device, and the device will pick up these tuberculosis particles, and
whatever
give a detection.
it is.
Yeah, they're quite wary of mentioning what it is. I think it's just a race now between different e-noses.
Yeah, different e-nose companies.
Yeah, exactly.
Yeah.
So yeah, and so the rats are also used for detecting landmines and such, but there aren't really e-noses for that. But we know today, for example, that dogs can detect low blood sugar, oncoming epilepsies and stuff like that.
Yeah.
So there's a lot about smell that we don't know.
Obviously, really. I mean, but I think they these e-noses also could smell cancer.
Yeah,
Right,
exactly.
yeah.
And also, for example, cats and bees has been known to be able to detect stuff like this. So it's really interesting. There's a whole new world that we don't know.
Don't pay attention to
Yeah,
that one.
there's people playing snooker downstairs.
Snooker?
Yeah, snooker.
What? Is that how you call it? Snooker?
Yeah, snooker. There's billiard there's different. Those are different kind of games.
Okay. Anyway, we're off topic. Okay, so.
But it's really important with the tuberculosis detection. In richer countries, it's maybe forgotten, but around 1.7, 1.5 million people die each year from tuberculosis.
Yeah.
And also people affected with HIV are much more prone to die and get the disease, about 20 times more likelier.
Yeah, and that is because obviously, I mean, a lot of people know this by now, that the HIV virus infect our body and then attacks our own immune system.
Exactly.
So our immune system decreases its not production.
Its detection rate or activity. The HIV virus, which we mentioned in the last episode, is a form of retrovirus. So they rewrite the DNA in T regulatory cells, amongst many. And T regulatory cells is like the
it's like a
conductor of the immune system.
It kind of like a captain of the immune system.
They can
kind of gives orders to other immune cells.
Exactly. And also there's what's it called? It's a lot of discrimination for people with HIV.
Oh yeah,
and so people are really afraid to get the diagnosis tuberculosis. So it's really important to have a good detection method.
Hold on, give that another spin.
People with HIV are more prone to tuberculosis. So people think that all people with tuberculosis has HIV.
Oh, okay. So yeah, okay, I get it. So they think the local population, if they see someone with tuberculosis, they also kind of in their head like, oh, they have HIV as well, so I better avoid that.
Exactly. It's a stigma against those people. So about just 50% of the people with tuberculosis is diagnosed. So with better detection methods and also a bit more knowledge about it, it can help a lot of people.
Well, what do you know about that? So, anything else? What more is there to, know about this? I mean, is it a new thing or?
The e-noses.
Yeah, exactly.
It's quite new. It has been researched last like maybe a decade, maybe less. But with better electronics, smaller computers, phones, and everything, it will be more and more easier to do this, and also more and more cost effective because it's a really push to get electronics much more cheaper.
Yeah.
But as I said, there is detection methods now which has the accuracy of 95.5%. And so maybe in the future we can use Apopos rats, a microscope, and these e-noses as a combined method to ensure that no one gets missed.
And Popo was the rat, not
the organization.
Not the police.
No.
Organization was it?
Yeah.
Oh, I thought it was the name of the rats.
No.
What was the name?
Giant pouched rats.
Okay, sorry.
So Popo is an organization that works with rats for tuberculosis and mine detection.
Mine detection. Oh, great. I have just a little comment, I suppose you can say. You hear all this. I mean, like amazing techniques that can solve, all the diagnosis of tuberculosis and also, for example, cancer. And you hear this rumors of all these amazing techniques, but just never go. When will we reach to the point when we actually get it? And we're like, oh, okay, we got it. Yeah. Can you actually?
I mean, the e-nose for tuberculosis seems to be quite finished, but it has an accuracy rate and all that stuff. Yeah, but it needs to have a larger test group. It needs to have all these different loops. It needs.
So once again, it's. I mean, so they still have to go through another, evaluation of this new technique. I mean, obviously. A lot of good reasons why they have to take this long to get a new technique out. Obviously, we're talking about lives here, and cancer and tuberculosis that can cause death, obviously, and a lot of other damage to humans. And so obviously, there's a good reason for this long evaluation time. But just I don't know. It was just a small comment that I kind of
it's just like gene editing and everything is just it will come someday, but it never does. But maybe this seems quite near the future than gene editing, at least. I don't know if that says that much, but it's at least something.
Yeah,
I think. And also, as I said, these diseases tuberculosis and such are deadly, so there isn't much choice than to increase diagnostics. Because it's quite hard to reverse it once it has become advanced. Because the tuberculosis, it goes into the macrophages, which is a part of the immune cell. I mean, immune cell that eats normally bacteria, but this evades the macrophage eating processing and starts to replicate inside it, and it destroys the lung because it starts to get it becomes like lumps of these immune system cells. And also it can go to the bloodstream and up to your brain and muscles and all that stuff. So it's quite a ugly disease, so to speak. So I think it isn't any choice than to just start to research on these e-noses and all that stuff.
No, absolutely. I mean, so obviously a great contribution to solving this problem. Obviously, the diagnose, that is. So yeah. Oh God, what a depressing episode we have right now. I mean, do we? Anything else you would like to add to this subject? As you obviously have already seen, I have no idea about this, these e-noses and the popo, and the organization.
We can say try to get vaccinated for TB if you have that
Oh yeah,
possibility.
yeah.
If you are worried, go to a doctor. And I don't think
Have a rat smell you.
Have a giant pouch rat at your home. Contact that popo.
You can adopt one. So
you can adopt a rat, or buy it at dinner.
And if for you people who like, wait, what? You can buy it dinner for the rats? Yeah, you can actually do that. Well, it's
a dinner for two rats, so they can have dinner together.
Romantic. No, yeah, exactly. So you can actually, you can help the organization by sponsoring them by being by being
a donor
or father.
I don't know.
I know. You can sponsor them by buying, I don't know, these rats dinner, I suppose.
And also you can become a scientist and research on electronic noses.
Yeah, you can do that.
Or try to cure tuberculosis.
Yeah, no.
This isn't our iGEM project, unfortunately, but maybe a future iGEM project.
Oh, yeah, absolutely be. I mean, definitely be intriguing to look into this subject even more, as I said at the last episode, obviously. But we do have a lot of other subjects that we would like to get into, which is more connected to our project. At least I'm way more, sorry, Popo. Thank you so much for your research and your time, and I will be sure to buy your rats at dinner. But yeah, no, I'm really looking forward to these next episode we will be airing.
Yes, and also soon in the future we can give you more updates on our iGEM project and how it goes. Maybe we will become more at home at the lab and not just run around like a confused giant pouched rats.
Exactly, and be way more comfortable around these microphones, as you can obviously notice that it's so uncomfortable right now. But anyway, I think, do you have anything else to add?
No, nothing. Just thank you for listening, and please come back and listen again.
Please come back. So once again, thank you. Well, sorry, you said it. Thank you, and
Goodbye.
Goodbye.